All right, so last time we looked at the nature of life. We discussed what science is and is not. And then we went into talking about different types of data and what does it mean to think like a scientist. And then we finished that section up. And that would lead us into section 2, how scientists work. So as always, science begins with curiosity. You know that as a child, you were very curious about many things. You'd often ask questions, why, how, what does this do, what does this mean? Well, scientists do the same thing. They have this curiosity about the natural world, and from that curiosity, they start to develop questions. And those questions then become what we are going to test. So it's a process that involves those asking questions, making observations, so collecting data using our senses, using that data to make inferences. So why does something particular happen in this scenario? And then going about and testing. So the whole testing idea is to collect data, whether it be qualitative data or quantitative data, and that data is going to tell us a story. And that story is going to be based upon those data points, observational or numerical, and it's going to essentially be the facts that are going to support or reject the hypotheses. So scientists use a process called the scientific method to find answers. And you learn about the scientific method every year in a science class, and you often think of it as a linear process. You'll be introduced to it as a linear process here. So what does that mean? Well, basically, we know that a linear process is a sequenced order. Step 1, step 2, step 3. And although it's introduced to you in this format, please understand that science as a process isn't always following that sequence of asking a question, forming a hypothesis, designing an experiment, collect and analyze data, and drawing conclusions. Scientists are kind of all over the place in that process when they're trying to find the answers about the natural world. So what does it mean to ask a question? Well, in your textbook, there is an experimental design written up concerning Radi's experiment. And basically what Francisco Radi suggested was, how do new living things or organisms come into being? Well, that is a question that he was wanting to know the answer to. So does life just spontaneously occur, meaning it occurs out of nowhere, or does life come from other living things? So from that, he would develop a hypothesis, or from your question, you would then develop that hypothesis. And a hypothesis, although often referred to as an educated guess, is a lot more than just an educated guess. A hypothesis has some important components to it. A hypothesis 1 must contain your dependent and independent variable, and we'll talk about those a little bit later. It should be written in biology. We'll talk about writing them as an if-then statement. And that statement, what makes it a true hypothesis is that it's not just any type of statement, a statement such as, how are you today, or good morning. Those are statements. Statements that are developed in the hypotheses must be testable. So they have to be testable statements. And if you don't have a statement that is testable, then you do not have a true hypothesis. And we base our hypotheses off of that previous knowledge or experiences. So we take into consideration what we already know about that particular topic. And essentially, the hypothesis itself is going to answer the question of our experiment or the problem that we are seeking to find answers to. So once we have done those two things, typically the next thing to do would be to come up with a controlled experiment. And a controlled experiment is an experiment in which you're only testing one variable. You only want to change one variable at a time because you want to have, you want to be able to compare the results that you're seeing within both your control group and that of your experimental group. So in a controlled experiment, you typically have a control group where all things are going to be constant or stay the same. In your experimental group, you are going to manipulate something. That would be that one variable that you're going to change. That's what manipulation means. You're going to change one thing, and that one thing is going to give you data. So something's going to respond to that change. And that's where our two variables come into play here. The two variables would be the manipulative variable. The manipulative variable is also called the independent variable. And that is the variable that you are going to change in an experiment. Okay? So write that down. The independent variable is the variable that you will change in an experiment. Okay? The other type of variable in an experiment is referred to as the responding variable. The responding variable is also known as the dependent variable. And that variable is going to be observable or measurable data. And that observable or measurable data is going to change according to the change that you made in the lab. So the responding variable or dependent variable depends on the independent variable, that thing you are changing. We also have a third type of variable in the experimental design process, and those things would be your controlled variables. And I'll talk a little bit more about controlled variables as we go and practice this first lab, dealing with the scientific method. The next thing we want to do is record and analyze our results. Here's where we get into the habit of taking that raw data, whether it be observational or numerical based, quantitative or qualitative, and we're going to organize it into data tables. And we could even take that raw data from the data tables and create graphs. There are three types of graphs that we create in science. The most common form is a line graph. A line graph is used to show changes or trends over time. So be sure to write that down. A line graph is used to show changes or trends over time. You can also create a bar graph, if I can spell it correctly. A bar graph is also called a histogram. And we use a bar graph or a histogram to compare things. So bar graphs or histograms are used to show comparison. And then the third type of graph is called a pie graph, or sometimes referred to as a pie chart. And a pie graph or a pie chart is used to show parts of a whole. And we will discuss graphing in greater detail in an in-class activity. So you will create graphs and you'll learn how to interpret graphs, because that is an important skill in science. Also, when you record and analyze data, here is where you're also going to want to do statistical or mathematical calculations. So you're also going to want to do statistical or mathematical calculations, and those statistical or mathematical calculations are going to help you further support or reject the data. The data itself is going to be your story for when you're drawing up your conclusions. So it is very important that in the last step of the scientific process, which includes drawing conclusions, you use that data to show support or rejection of the hypothesis. You need to reference the data because that is going to provide the factual information to show that support or rejection of the hypothesis. If the hypothesis is supported, then you could go on and do further testing. And if you get continuous results that show support for that hypothesis, that could develop into something further. If the data rejects the hypothesis, then you simply go back and you first look for experimental errors. If there aren't any experimental errors, then you'd want to go back and just modify, come up with a different hypothesis. So you'd want to modify your hypothesis and then retest it and see whether the data will support or reject it. So in science, we often try to do experiments that are controlled, but not all experiments can be controlled. And examples are right here illustrated. If you're studying animal behavior in the wild, you really don't have any control over what that animal is going to do, so you really can't do a controlled experiment there. In science, especially in biology, we often run across ethical situations. Ethical situations such as this is where we might have issues between what we're trying to learn in science and its confliction with the values of the church. So sometimes ethical situations come into play where it hinders the process of science. And we will spend a little bit of time talking about some bioethical issues throughout the class. If you test more than one variable at the same time, that would not be a true controlled experiment. When you go to test more than one variable, you have nothing to compare what is causing that change in the experiment. For example, if you manipulate two things, one thing being A, the other thing being B, you can't decipher whether it was A or B that was causing that change when trying to compare it to your control group. So it is always critical to only test one variable at a time. And of course, money constraints. So money plays a huge factor in whether or not research can be taken to its completion. I know a couple people who, during their graduate research, working towards a PhD in science, basically made it so far in the process, and due to lack of funding, their research was cut off and they did not receive their PhD. So money plays a huge role in the types and amounts of experiments that we could do. So beyond a hypothesis, we also have scientific theories. A theory basically is developed after a hypothesis or many hypotheses have been tested and supported through repeated experimentation. So these all are supported through factual evidence that we have at that time. There are several theories that we'll study in science. We have the germ theory, gene theory, cell theory, the theory of evolution. These are all theories of biology. In science, the word theory applies to well-tested explanations that unify a broad range of observations. So one of the big ones being the cell theory, we say that all living things are made of cells. We know that cells are the basic unit of structure and function of all life, and that new cells come from pre-existing cells. Those were experiments, all those statements of that theory were at once hypotheses that were tested and retested again to provide that factual evidence, and collectively those hypotheses have been developed into a scientific theory. The other thing that we have in science is a scientific law. Scientific laws